Welcome to the latest deep dive of the Worm Podcast. And look, we know you, our listeners, are already intimately familiar with our favorite nematode.
Oh, yes, C. elegans.
Right. So, because you already know the basics, we are totally skipping the basic biology today. We know, the anatomy, the life cycle, the whole deal.
Yeah, we don't need to rehash that. We can just jump straight into it.
Exactly. We are jumping straight into a frankly mind-bending question today. And it's this. Can a mother's diet physically alter the fundamental cellular machinery her babies are born with?
It is a phenomenal biological puzzle, honestly. And to pull it apart, we're looking at a really fantastic paper.
Yeah.
This is from Pradhan, Tobin, and their team.
Yeah, published in PLOS Biology.
Yeah, PLOS Biology, April 2026. And they essentially set out to map how dietary restriction in a mother bridges the generational gap. Like, how does it control the ribosome abundance of her offspring?
Which is huge.
Right, and how that early provisioning dictates the baby worm's development later on.
Because that fundamentally challenges the assumed boundaries, between a parent's lived experience and an embryo's starting state. We're going to get deep into the molecular weeds on this one.
Oh, definitely.
But before we do, a quick favor for you listening. Please take a second to follow, like, comment, and share this deep dive. I mean, it's how we keep the worm community growing.
Yeah, we really appreciate it.
And hey, if your lab has research you'd like us to feature, Dr. Viren Chauhan's contact details are right down in the show notes. Just reach out to him.
We always want to see what you're working on. But to set the stage for this specific paper, we really have to look at the growth law.
Right.
We see this incredibly clearly in microbes. Basically, a cell's ribosome expression scales linearly with its growth rate.
So if the environment is rich and the cell is dividing rapidly, it just cranks out ribosomes to meet that massive demand for protein synthesis.
Exactly. But if the environment is poor, keeping all that translational machinery active, that is a massive metabolic drain.
It's just too expensive.
Yeah, it is. And multicellular animals followed this too. So the researchers showed that when C. elegans mothers face dietary restriction...
And in this case, it was a 5-fold dilution of their bacterial food, right?
Yeah.
So it takes them, 5 days to reach adulthood instead of 3?
Yep, 5 days instead of 3. And when they do that, they downregulate their ribosomes.
I mean, they match their internal factory capacity to the external supply chain. That makes perfect sense for the mother's own somatic cells.
Totally logical.
But where I get tripped up is the transfer to the next generation. Because, we have the Weismann barrier.
Right, the classic Weismann barrier.
The germline is physically and molecularly sequestered from the somatic cells very early in development. I always think of it like a smartphone factory reset.
That's a good way to look at it.
Yeah. If a mother worm has a terrible environment, sure, her somatic cells adapt. But when she provisions an embryo, she's supposed to hit the biological factory reset button.
The new user, the baby, shouldn't inherit her scrambled settings.
Exactly. The physical reality of that reset is intense. The germ cells are shielded, their epigenetic marks are scrubbed, and the proteome isn't just passively bleeding over from the mother's gut or whatever.
No, it's not. And to see if any proteins actually survive that reset, the research team deployed TMT proteomics.
Tandem mass tagging.
Yeah.
So they're taking the protein extracts from the progeny of well-fed mothers and then the progeny of restricted mothers and chemically attaching these tiny isobaric tags.
Which act like microscopic barcodes, basically.
Wow.
And because those tags have slightly different molecular weights that only reveal themselves when they're fragmented inside a mass spectrometer, the researchers can just mix the samples together.
Run them through the machine simultaneously.
Exactly. And they get an incredibly precise relative quantification of over 6,400 distinct proteins.
Which is just a staggering snapshot of the total cellular inventory. And looking at that data, the factory reset analogy largely holds up.
It does.
Out of those 6,400 proteins, the vast majority of the proteome changes triggered by the mother's starvation simply do not pass to the offspring. The slate is wiped clean.
Almost entirely clean. There is one massive glaring exception sitting right in that proteomics data.
The ribosomes.
The ribosomes. Out of the 6,400 proteins measured, the team detected over 140 ribosomal proteins, and nearly all of them remain significantly downregulated in the newly hatched L1 progeny.
Just to be clear, these are the progeny of the dietarily restricted mothers.
Right, exactly. The actual protein factories themselves bridge the gap.
Okay, but I mean, proteomics involves grinding up a bunch of worms and looking at the bulk bulk average. A skeptic could argue that, maybe the extraction process favored certain proteins,
Yeah.
or it's just a statistical artifact.
And the researchers anticipated that exact critique, so they validated the mass spec data with live imaging.
Oh, nice.
Yeah, they used C. elegans strains with endogenously inserted fluorescent tags. They basically used CRISPR to insert the sequence for M. cherry directly into the genome next to the RPL34 ribosomal gene.
And GFP next to the RPL29 gene.
Yep. So the worms are transcribing and translating these glowing tags under their own native regulatory control.
Not from some, artificially pumped up extrachromosomal array.
Exactly. You can literally just look at the live L1 worms under a microscope and measure the fluorescence intensity to see how many ribosomes are actually there.
And what did they see?
The fluorescence data mirrored the proteomics perfectly. The L1 progeny from restricted mothers were visibly dimmer. The protein factories were undeniably down.
But, okay, if I'm playing devil's advocate here, a starving mother produces smaller eggs, right?
Yeah.
Which hatch into physically smaller L1s. So if the entire worm is tiny, of course there are fewer total ribosomes. Are we just looking at a byproduct of the baby being miniature, or is the actual concentration, like the density of ribosomes per cubic micron of worm, actually lower?
That is a great question. That distinction between absolute abundance and volumetric concentration is the crux of the whole issue.
Right.
The researchers carefully size-normalized their fluorescence and proteomics data to account for the reduced volume of the offspring.
Okay.
Even after factoring in the smaller physical size, there was an 11% reduction in the actual concentration of ribosomes at hatching.
Wow. An 11% drop in the fundamental density of the translational machinery.
Yeah.
But I also have to wonder, is she just indiscriminately packing less of everything complex into the egg to save energy?
Yeah.
Like maybe all the large multi-protein complexes are downgraded?
Well, they tested that too. They looked at the proteome, which is the cell's primary protein degradation complex.
The trash compactors.
Yep, it's another massive, metabolically expensive piece of machinery. So they tagged a proteome subunit, RPN9, with M. scarlet.
And what happened?
In the starving mothers, the proteome was heavily downregulated, as you'd expect, but in the offspring,
Let me guess.
the proteome levels reset perfectly to normal.
No way. So she intentionally hits the factory reset button on the recycling centers, but manually overrides the reset for the manufacturing centers.
Exactly. It's a highly targeted intergenerational transfer of state.
Which forces us to look at the consequences for the baby worm. I mean, they hatch with an 11% deficit in their protein factories. What happens if that ribosome-deficient L1 is suddenly placed in an environment with abundant food?
Right, like they get dropped onto an agar plate just paved with E. coli.
The all-you-can-eat buffet.
Yeah, the buffet. So the researchers tracked individual worms in these arrayed agarose microchambers, measuring their exact developmental timing. And when the progeny of restricted mothers were put in that nutrient-rich environment, they hit a significant developmental roadblock.
How significant?
There was a 2.7-hour delay just to get from the L1 to the L2 molt.
Wow.
And that was accompanied by a markedly slower initial growth rate compared to babies from well-fed mothers.
Well, that makes sense mechanistically, right? The environment is demanding rapid cell division and massive tissue growth, but the worm just doesn't have the hardware to process the demand. It's like trying to run a heavy, modern piece of software on an outdated processor. Like, you have all the data you need to run the program, the food, but the system just lags and stutters because it can't physically compile the code fast enough.
That's a great analogy. But the hardware isn't fixed permanently. Over the course of that delayed L1 stage, the restricted progeny are furiously synthesizing new ribosomes.
They are building the very factories they need to process the food.
Exactly. And the imaging shows that by the time they finally hit the L2 stage, their ribosome concentration catches up to the well-fed control worms.
So they recover.
Completely. From L2 onward, their growth trajectories are practically identical.
They download the hardware updates in the background, and once the installation is done, the software runs smoothly. But, here is the major trap with this kind of observational data.
Oh, causality.
Yeah. A starving mother passes on a lot of different stress factors. Maybe the egg has altered lipid stores, or different epigenetic methylation patterns, or altered mitochondrial function. How can we definitively point to the ribosomes and say, that 11% deficit is the direct physical cause of the 2.7-hour delay?
Right, because correlation isn't causation. Proving causality there requires decoupling the ribosome deficit from the mother's starvation entirely.
You'd have to create a ribosome-deficient baby from a completely well-fed, unstressed mother.
Exactly. And to do that, the team utilized auxin-inducible degradation, or the AID system.
Ah, the AID system. This is
Yeah.
one of my favorite tools that we've hijacked from plant biology.
It's incredibly elegant, isn't it?
It really is.
They genetically tagged a single ribosomal protein, RPS26, with an AID tag. On its own, the worm cells completely ignore that tag,
Right.
but the researchers also introduced a plant transport protein called TIR1 into the worms. When you expose these worms to the plant hormone auxin, the auxin acts as a kind of molecularly double-sided tape.
It binds the AID tag on the ribosome to the TIR1 protein.
Yeah, and TIR1 physically drags the tagged ribosome right into the worm's own cellular trash disposal system, the ubiquitin proteasome pathway, just shredding it.
It is a highly specific remote-controlled destruct switch.
Exactly that.
Mhm.
So the team restricted the expression of that TIR1 protein, so it was only active in the mother's germline and the early embryos.
Okay, so the mothers aren't starving. They have all the E. coli they can eat.
Happy, well-fed mothers.
But they put them on plates containing auxin, and the molecular glue does its work. The auxin is reaching into the developing oocytes and surgically destroying a fraction of the ribosomes as they are being packed.
Exactly. And when those specific eggs hatched, the L1s exhibited the exact same phenotype.
No way.
Yeah, they had the delayed L1 to L2 molt, they struggled with initial growth, and they eventually caught up. It perfectly mimicked the progeny of actual starved mothers.
Because it's isolating the variable.
Yeah.
It wasn't lipid shortages or general stress. It was purely a mechanical limitation of translational capacity.
Pure hardware limitation.
That brings us to the biggest mystery of the paper, though. We know the mother intentionally overrides the Weismann barrier to restrict ribosomes. We know that restriction causes the lag. But how does a hungry mother's body physically communicate with her germline to make that adjustment?
So the researchers targeted the mTORC1 signaling pathway, which is, the master metabolic sensor in eukaryotes.
Right.
It integrates signals about nutrient availability, energy status, and oxygen levels. And its primary job is regulating cell growth and, crucially, ribosome biogenesis.
Okay, so to manipulate this, the team focused on RIGA1, right? A protein that acts as an essential activator for mTORC1.
Exactly. If you knock down RIGA1, you blind the mTORC1 complex.
The cell might be swimming in nutrients, but the master regulator thinks it's starving and shuts down ribosome production.
Yeah, and when they ubiquitously depleted RIGA1 in the mother, so turning it off in every single tissue, it perfectly phenocopied dietary restriction.
The offspring hatched with reduced ribosomes and grew slowly.
Right. But ubiquitous depletion is a blunt instrument. To find the specific origin of the signal, they turned to tissue-specific promoters to knock down RGA1 in isolated organs.
This is where we figure out who is talking to whom.
Exactly. First, they used the myo2p promoter to deplete RGA1 exclusively in the pharynx.
The worm's neuromuscular feeding tube, the first point of contact with the food environment. And what happens when only the throat thinks it's starving?
The mother produced offspring with the signature intergenerational ribosome reduction.
Okay, but if I'm the germline, I shouldn't necessarily trust just the throat, right? The throat thinks we're starving. The whole mother worm is going to be physically stunted because she isn't eating enough.
Sure, that makes sense.
Couldn't the germline just be sensing that the body housing it's generally small and unhappy and just reacting to that overall state? Did they check other tissues?
They tested that exact alternative. They used the Cool10P promoter to deplete RGA1 specifically in the epidermis, the worm's skin. No. Disrupting mTORC1 in the epidermis profoundly stunts the mother's overall growth. She becomes physically tiny, just like the mothers with the pharynx depletion.
So you have two populations of mothers.
Huh.
Both are physically stunted. Both have localized mTORC1 disruptions.
But here is the critical divergence. The mothers with the disrupted epidermis did not produce ribosome-deficient offspring.
Wait, really?
Their babies hatched with normal ribosome levels.
That is wild. That completely rules out the idea that the germline is just reacting to the mother's overall size or a general state of, smallness.
Yep.
The germline is listening to a highly specific direct message. The feeding tube is actively communicating with the reproductive system.
It highlights the pharynx as the primary environmental sentinel. And it makes physiological sense. It's the organ physically grinding the bacteria. When the pharynx senses a drop in nutrients, it transmits a signal likely via neuropeptides or circulating metabolic factors in the pseudocolum, that travels down to the germline and essentially says, the external environment is deteriorating. Scale back the translational hardware in the next batch of embryos.
It's an incredible piece of cross-tissue coordination. But looking at it from an evolutionary perspective, it feels risky.
How so?
Well, if a mother is starving, she packs the egg with 11% fewer ribosomes. If that baby happens to hatch onto a rich patch of bacteria, it's severely handicapped. It loses almost 3 hours of critical early growth time just trying to build the hardware it should have been born with.
That's true.
In a competitive environment, 3 hours of lag could be lethal. Why wouldn't evolution just pack every egg with max ribosomes just in case? Is this intergenerational reduction a bug, or is it a deliberate feature?
The authors argue it is strongly a feature. We have to view this through the lens of predictive adaptive response.
Okay, predicting the future based on the present.
Right. Environments in nature usually have some temporal continuity. If a mother is starving, the statistical probability is that her baby is going to hatch into that exact same starved environment.
Oh, I see. It's like packing a backpack for an expedition.
Yeah.
If the guide tells me we are going on a grueling, low-resource hike, I'm going to pack ultralight.
Yeah.
If I suddenly get dropped into a sprint, I'm going to fail because I don't have the gear.
Exactly.
But in the scenario I was actually predicting, the light pack is perfect. I didn't waste energy hauling heavy equipment I couldn't use.
And ribosomes are incredibly heavy equipment. The transcription of ribosomal RNA and the translation of ribosomal proteins consumes a massive fraction of a cell's total energy budget.
They're huge.
For a mother worm who is actively starving, continuing to manufacture massive quantities of ribosomes to stuff into her oocytes, that's a metabolic death sentence.
So by lowering embryonic ribosome deposition, she drastically reduces her own metabolic cost of reproduction.
She saves her own life, essentially. And she perfectly equips the offspring for the reality they are facing. Now, to test this computationally, the researchers faced a physical hurdle. You can't easily keep newborn worms totally starved in those microchambers and still image them effectively.
Because they just die or wander off?
Pretty much. So they used their agenetic toolkit again to mimic starvation. They used the RGA1 aid system in the offspring to artificially suppress their ability to process nutrients.
Oh, clever.
Right. Essentially forcing the baby worms into a physiological state of starvation regardless of the food around them.
They enforce the slow hike scenario.
Exactly. And under those enforced slow growth conditions, the growth disparity between the babies of well-fed mothers and the babies of starved mothers nearly vanished.
Because they're both forced to grow slowly anyway.
When the environment, or in this case the internal physiology, doesn't permit rapid growth, having fewer ribosomes at birth isn't a handicap at all. It's the optimal state.
So it's a massive evolutionary win-win, provided the environment remains consistent. The mother conserves critical energy during a famine, and the baby isn't burdened with an overbuilt, metabolically expensive factory. It doesn't have the supplies to run.
Right.
It's only viewed as a handicap when the environment suddenly flips to abundance, which is, I guess, rare enough that the evolutionary math favors the energy savings.
Exactly. It reveals a profound layer of intergenerational plasticity. The Weismann barrier protects the genetic code, sure, but it is selectively permeable to metabolic realities.
The hardware you inherit from your parents isn't just a static blueprint. It ships with the dials already adjusted based on the sensory data your mother collected before you were even a zygote.
It's fascinating.
Her throat sensed a famine, and your cells were pre-calibrated for scarcity.
Which forces us to stop thinking about inheritance purely in terms of DNA sequence. The inherited proteomic state dictates the functional capacity of that DNA in the critical early hours of life.
And, well, that leaves me with a final, slightly provocative thought to chew on.
Oh boy, let's hear it.
If a mother's environment can downregulate the starting baseline of ribosomes by 11%, what happens when we stack that across generations? If generation 1 is restricted, and generation 2 hatches with a deficit, grows slowly, and remains restricted, does generation 3 hatch with a 20% deficit?
That's a million-dollar question.
Right. Could subtle dietary shifts, compounded over multiple generations, theoretically lock a population into a permanent slow-growth state just by compounding this initial ribosome deficit over and over? Or is there a biological hard floor to this loophole in the Weismann barrier, where the organism simply refuses to scale down any further? How deep does this molecular memory actually go before the system breaks?
The existence of a biological floor and how epigenetic marks might interact with this direct protein provisioning over multiple generations, that is exactly the kind of frontier this paper opens up. It's a question the field will undoubtedly be chasing next.
And I can't wait to see the data when they figure it out.
Well, that brings us to the end of today's deep dive. Thank you so much for listening. And remember, you can help us keep the Worm podcast community thriving.
Please do.
Please follow, like, comment, and share this deep dive. And don't forget to check the show notes to contact Dr. Viren Chauhan if you have a paper you want us to feature next. Until next time, keep digging into the data.